Hello students, hi. Now the last part, but this is important questions from reproduction in flowering plants, revision questions and answers. Describe the process of development of dicotyledonous embryo. The process of development of mature embryo from diploid zygote is called embryogenesis. Means that developing mature embryo from diploid zygote. They'll ask what is embryogenesis, then you can write this. After fertilization, the zygote of ovule divides transversely into two cells, a small apical cell and a large basal cell. The basal cell lying towards the micropyle divides in one direction into a row of cells called a suspensor. The diagrams chart is already there in textbook, and at the end of this question answer you can observe those cells which are their growth. Those you have to refer for these. The basal cell lying towards the micropyle, which is down, divides in one direction into a row of cells called a suspensor. It pushes the developing embryo into the endosperm for absorption of nutrients. So the apical cell located towards antipodal end of zygote undergoes two vertical divisions and one transverse division to form the embryonal mass. The cluster of cells, which are ladder-like structures, those. The cells of the embryonal mass divide repeatedly and various parts of the embryo are formed. The anterior cells of the embryonal mass, which is upside going, and it grows as a shoot system or stem, those are called plumule, and two cotyledon, as I already discussed about dicots, which are plumule and radicle. Radicle is the root, plumule is the shoot. So the main part of the radicle and the hypocotyle are formed from the posterior embryonal mass of cells. See, this is the diagram for that. Degenerating synergids, zygote, which is diploid, and you can say primary endosperm nucleus, which is triploid, which is even responsible for the coconut plant. Coconut gets the endosperm inside, how the water will come inside coconut. This is the reason also, primary endosperm nucleus. So primary endosperm cell, PEC it is called. So when they ask reasonable or logical questions, you can write for coconut getting water. And degenerating antipodal cells, which are three down. So zygote, then two celled, then another is like lady's hair, which is globular embryo. Then another one is next cell is heart shaped embryo, and the last one matured embryo, which is showing here in diagram plumule and radicle and cotyledon. So trace the events that would take place in a flower from the time the pollen grain of the same species falls on the stigma up to completion of fertilization. So the pollen grain develops on the stigma, stimulated by the secretion of stigma. The intine grows out as a protuberance, means protuberance means come out like pimple through one of the germ pore. This outgrowth continues to grow as a pollen tube. The nucleus of the vegetative cell moves into it, followed by the generative cell. The generative cell divides into two male gametes and moves to the tip of the pollen tube. The pollen tube secretes enzyme to digest the tissues of the style and enters the ovule through the micropilar end and discharges the two male gametes into one of the synergids of the embryo sac. One of the male gametes fuses with the ovum to form a zygote. This process is called syngamy. The other male gamete fuses with the secondary nucleus to form primary endosperm nucleus. See, when they ask what is syngamy, you can say male gamete combines with the zygote with the ovum to form a zygote. And this is very important question for 1 mark, even for NEET. So the process is called triple fusion also, since there are two fusions, syngamy and triple fusion inside the ovule during fertilization. It is known as, because of two fertilizations, it is called double fertilization. This is very important. So please learn the concept of syngamy, triple fusion and double fertilization and primary endosperm nucleus. So this is the diagram for that, which is showing in stigma where pollen tube enters through the down, which is darker color, longitudinal section of a flower showing growth of the pollen tube. The pollen tube enters into ovule, that is synergid and egg cell polar nuclei antipodal by small cells. Those are forming, it is showing in this diagram. And the male gametes, and further it develops male gametes through vegetative nucleus filiform apparatus. The further division, central cell, egg nucleus, plasma membrane. This diagram is important. So the next question is, describe the structure of a typical embryo sac found in flowering plant. Why is it generally referred to as monosporic? A typical embryo sac is 8 nucleated, 7 celled. Even they will ask not only a typical embryo sac diagram, they will ask sometime 8 nucleated, 7 celled embryo sac diagram, or explain 8 nucleated, 7 celled embryo sac. Then you have to write this. So whatever cells, 6 of the 8 nuclei become surrounded by cell wall, as I have shown already in earlier diagram, that cell wall and organized into 2 cells. 2 of the nuclei, called polar nuclei, remain in the center of the large central cell. In the center, polar nuclei will be there like 2 dots. 3 cells are grouped together at the micropilar end, forming egg apparatus of this. One is a female gamete and 2 synergids. The synergids have special cellular thickening to form down, which is filiform apparatus like finger-like projections shown in the diagram at the micropilar tip. So 3 cells are grouped at the chelagel end. They are called antipodals. The large cell in the center of the embryo sac is the central cell. Later 2 polar nuclei in the central cell fuse to form diploid secondary nucleus or endosperm nucleus. Thus the embryo sac of flowering plants is 8 nucleated, 7 celled maturity. This type of embryo sac is called monosporic because it is formed only from 4 megaspores. So with a neat label diagram, describe parts of a typical angiosperm ovule. Even this is one example of the typical angiosperm ovule. There will be many questions are orthotropous, anitro, based on the shape of the ovule, how it is, name of the ovule written. This is the angiosperm ovule, the general one, which is the parts one by one, the purpose of those function I will describe. The funicle, which is the first stalk like flower, stalk flower, it is a stalk-like structure which represents the point of attachment of an ovule to the placenta of the ovary. Inside the flower, funicle will attach to the ovary. This is a umbilical cord between the mother and placenta. That placenta, this is funicle, like that connection. So hilum, the next part is hilum. It is the point where body of ovule is attached to the funicles. So they will ask for this, what is hilum, what is punicle, for 1 mark question. So please do remember. So next one is integuments. These are like for corn, sweet corn or corn layers like that. This one, they are outer layers surrounding the ovule that provide protection to the developing embryo. This is like 2 layers, outer integument, inner integument. There are layers which is protecting the center embryo. So micropyle is the narrow pore which is formed by the projection of integuments. Next to the integuments, this micropyle will be there, which is pollen tubes enters the ovule at the time of fertilization through this micropyle only. So next one is nucellus, not nucleus. This is nucellus. There is a difference between nucleus and nucellus. So nucellus is the mass of parenchymatous tissue which is surrounded by integuments from the outside. So the nucellus provides nutrition to the developing embryo. The embryo sac is located inside the nucellus. Shalaja. It is based swollen part of nucellus from where integuments originate. This is about angiosperm ovule. Diagram is important and the parts you have to write, and description for 5 marks you have to write all these. So the next question is differentiate between microsporogenesis and megasporogenesis. The process of formation of microspores from pollen mother cell through meiosis is called microsporogenesis. Production of microspores especially. The production of megaspores through meiosis from megaspore mother cell. This is microspore mother cell is MMC. This is MGMC. Megaspore mother cell, MGMC in small abbreviation. Okay. So the next point of microsporogenesis is pollen grains are formed at the end of this event, occurs inside pollen sac of anther. But whereas in megasporogenesis, egg is produced inside the embryo sac at the end of this event. What is meant by monosporic development of female gametophyte? So the female gametophyte, other name is embryo sac, develops from a single functional megaspore. This is known as monosporic development of the female gametophyte. In most flowering plants, a single megaspore mother cell present at the micropylar pole of the nucellus region of the ovule undergoes meiosis to produce 4 haploid megaspores. Later, out of these 4 megaspores, only one functional megaspore develops into female gametophyte, while the remaining 3 degenerates. So, the next question is, what are chasmogamous and cleistogamous flowers? There are nothing but what are open flowers and closed flowers. So, can cross-pollination occur in cleistogamous flowers? Give a reason for your answer. So there are 2 types of flowers present in plants, namely oxalis, which is a weed generally grown wherever soil is there, with the 4 leaves. Viola, chasmogamous and cleistogamous flowers. Chasmoga have exposed anthers, stigma similar to the flowers of other species. So cross-pollination cannot occur in cleistogamous flowers. This is because cleistogamous never open. These are closed flowers. The anther and stigma lie close to each other in these flowers. Hence only self-pollination is possible in these flowers. If it is open, then cross-pollination will be possible. So when you observe the flowers, you can see which is a closed flower and which is open.